The purpose of Bio-Rad Explorer's P-Glow Bacterial Transformation Kit is to introduce students to the basics of genetic engineering, gene regulation, and the central framework of biology: DNA, RNA, protein, trait. Students will take a gene from a bioluminescent jellyfish and transform bacteria, making them glow green from expressing green fluorescent protein, GFP. Number 1. Label one closed microtest tube plus P-Glow and another minus P-Glow. Label both tubes with your group's name and place them in the foam tube rack. Number 2. Open the tubes and using a sterile transfer pipette, transfer 250 microliters of transformation solution, calcium chloride. Number 3. Place the tubes on ice. Number 4. Use a sterile loop to pick up a single colony of bacteria from your starter plate. Pick up the plus P-Glow tube and immerse the loop into the transformation solution at the bottom of the tube. Spin the loop between your index finger and thumb until the entire colony is dispersed in the transformation solution, with no floating chunks. Place the tube back into the tube rack in the ice. Using a new sterile loop, repeat for the minus P-Glow tube. Number 5. Examine the P-Glow plasmid DNA solution with the UV lamp. Note your observations. Immerse a new sterile loop into the plasmid DNA stock tube. Withdraw a loopful. There should be a film of plasmid solution across the ring. This is similar to seeing a soapy film across a ring for blowing soap bubbles. Mix the loopful into the cell suspension of the plus P-Glow tube. Close the tube and return it to the rack on ice. Also close the minus P-Glow tube. Do not add plasma DNA to the minus P-Glow tube. Why not? Number 6. Incubate the tubes on ice for 10 minutes. Make sure to push the tubes all the way down in the rack so the bottom of the tubes stick out and make contact with the ice. Number 7. While the tubes are sitting on ice, label your 4 agar plates on the bottom, not the lid, as follows. Label one LB amp plate plus P-Glow. Label the LB amp era plate plus P-Glow. Label the other LB amp plate minus P-Glow. Label the LB plate minus P-Glow. Number 8. Heat shock. Using the foam rack as a holder, transfer both the plus P-Glow and minus P-Glow tubes into the water bath set at 42 degrees Celsius for exactly 50 seconds. Make sure to push the tubes all the way down in the rack so the bottom of the tubes stick out and make contact with the warm water. When 50 seconds have elapsed, place both tubes back on ice. For the best transformation results, the change from the ice, 0 degrees Celsius, to 42 degrees Celsius and then back to the ice must be rapid. Incubate tubes on ice for 2 minutes. Number 9. Remove the rack containing the tubes from the ice and place on the bench top. Open a tube and, using a new sterile pipette, add 250 microliters of LB nutrient broth to the tube and re-close it. Repeat with a new sterile pipette for the other tube. Incubate the tubes for 10 minutes at room temperature. Number 10. Tap the closed tubes with your finger to mix. Using a new sterile pipette for each tube, pipette 100 microliters of the transformation and control suspensions onto the appropriate plates. Number 11. Use a new sterile loop for each plate. Spread the suspensions evenly around the surface of the agar by quickly skating the flat surface of a new sterile loop back and forth across the plate surface. Number 12. Stack up your plates and tape them together. Put your group name and class period on the bottom of the stack and place the stack upside down in the 37 degrees Celsius incubator until the next day.
Subscribe to the Bio-Rad Explorer channel and you'll discover enriching and fun biology and life science videos. Watch what you'll explore. Subscribe now.